everyone, and welcome to today's webinar titled ZoomAb Antibodies, a Novel Recombinant of Antibody Technology. This webinar is part of the Cancer Research and Oncology virtual event. And I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by MilliporeSigma. For more information about MilliporeSigma, go to www.sigmaaldrich.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on the Ask a Question box located on the far left of your screen. As a reminder, the presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Wayne Speckman, Head of Antibody Development. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Speckman, welcome. You may now begin your presentation, sir.
Hello, this is Wayne Speckman. I'm from MilliporeSigma. I'm an R&D scientist, and I'm going to be presenting a new technology called ZoomAb today. It's a recombinant antibody technology. So just a quick agenda. So obviously, I'm just going to spend a little time talking about antibodies, make sure we're all on the same page. I'll dive into ZoomAb and what exactly these recombinant antibodies are and how they work and how they're developed. I'll kind of then shift into talking about our entire portfolio of antibodies, and then finally finish off with some resources. So antibody overview. So as many of you are probably aware, antibodies have become an important research tool for scientists. Researchers and clinicians, use them to identify and isolate specific molecules. So the antibodies are very specific, targeting a very particular molecule typically. So they're used for identification of target molecules. They're utilized for location of target molecules. Often these target molecules are markers for a cell type or even a tissue type. They can be markers for disease or physiological states. And then sometimes antibodies can even be used for purification or concentrating a particular target. So there's a lot of functionality and uses for antibodies. Antibodies are kind of broadly broken up into two types, polyclonal and monoclonal. So polyclonals, shown on the top, are a mixture of many different antibodies. These antibodies are non-identical because there are many different B cells that are producing each antibody. So each different type of antibody is produced by a different specific B cell. And so they detect multiple targets and epitopes. Okay. In contrast, there's the monoclonals, which is just one single type of antibody. So they're all the exact same sequence of amino acids, and they're identical because they're all produced from one B cell or a clone from a single parent cell of B cells. So they're all exactly the same, and obviously the difference is they're going to be detecting hopefully one epitope, and they all the specificity would be the is produced from those particular B cell. So there's advantages and disadvantages, and so we're kind of going to go into some of these for polys and monos. So polyclonals, they're easy to generate, very cost effective. You simply immunize an animal with an immunogen and pull out the serum, and you're going to have a polyclonal mixture. There's going to be lots of antibodies in that mixture, hopefully most of them for the particular target of interest, but that animal also is going to have antibodies to anything that it's been exposed to. The polyclonals might have multiple epitopes on the same protein, so one protein might be targeted by numerous antibodies. And sometimes this is good because it can lead to more robust signals in some immunoassays. This sometimes is helpful, especially with proteins expressed in low levels, to get a stronger signal in testing. Polyclonals are often compatible with a broader range of applications, and sometimes there's higher flexibility in antigen recognition because of being able to recognize multiple epitopes. In contrast, monoclonals, different clones of antibodies can be generated to different epitopes. So, you can have different antibodies that only recognize a particular epitope because the clone that was selected that antibody is selected because it only detects one epitope. The hybridoma serves as an infinite source of the same antibody. So once that hybridoma is produced and identified and isolated, it's kind of an infinite source. If it's screened properly, there can be minimal background and cross-reactivity. So if the antibody that's selected from a hybridoma is very clean, that is certainly helpful for monoclonals and tends to be a bonus that's different from polyclonals. And because they're producing only that one antibody, you get high homogeneity, consistent, and reproducible results. And then there's less batch-to-batch variation because it's all produced by one clone. So some of the challenges for polyclonals, obviously the animals have a certain lifespan, and that limits the amount of polyclonal material that can be produced. If the animal dies prematurely, that can also lead to less material. So there's definitely a lifespan and a limited amount that's produced. The other thing is over time, as the animal is exposed and re-exposed through boost, the bleeds that are produced may give different results. The animal may have a lower titer, giving thus less signal, or it may even completely change and lose the ability to produce antibody against the target of interest. So bleeds over time with one organism may change. This obviously causes greater batch-to-batch variability. You might get non-specific antibodies, again, because this is polyclonal. You can have antibodies that are recognizing multiple different targets, and this can lead to background signal. And immunization of a new animal with the same antigen, it may lead to different epitopes. So even if that original material that was great was produced, when you try to make it again, there's no guarantee that that additional organisms that are immunized with the exact same immunogen are going to produce the exact same antibodies. Again, they're polyclonal, they're different, so there's no guarantee that you'll get the same. And then there's shared epitopes on different proteins. So you might get background because the epitope that's recognized with one is also recognized on a second protein. So some challenges for monoclonals, sometimes limited working applications. So some monoclonals just only work in a particular application or two, and that's obviously got to be tested to be verified, but that can be an issue. They are susceptible to loss of epitope because it's only recognizing one epitope. If that epitope gets modified or changed or affected, you may lose the actual detection in a test. Sometimes there's higher specificity limits with multiple species. One of the big issues that can occur with monoclonals is hybridoma cell line drift, which means over time either the expression changes or the sequence changes, and the cell line itself, which is assumed to be exactly the same from the very beginning to months and years later, may not be exactly the same. And then obviously contamination or loss of the cell line would be a huge issue. If there's contamination and the cells are dead, there is no bringing that back. You'd have to completely restart it, or at least hope that you had other vials of cells that are somewhere else, stored somewhere else, that are still okay. So one of the great things is that sort of the best of both worlds is to actually use recombinant monoclonal antibodies. And that's what ZooMabs are, and we'll get into that in more detail. But recombinant antibodies are manufactured by cloning antibody genes into expression vectors, which are subsequently introduced into expression systems to provide animal-free and even preservative-free antibody production. So there's a lot of benefits. No antibody sequence mutations, high batch-to-batch consistency at the same exact process is used in terms of making them and purifying the recombinant antibodies. Significantly reduced production time. You don't have to wait for an animal. You literally are doing it in culture, so it's very quickly in terms of the turnaround time. There is increased reproducibility and control because the conditions from one batch to the next batch are exactly the same. There's zero or very minimal variation, so that the reproducibility is excellent. It's easy to develop humanized or alternate versions where you mutate some of the sequence if needed. If there's a poor expression, you can replace a tRNA that's a low quantity or low amount with a higher tRNA to get better expression. As I mentioned before, it's animal-free technology, so once the antibody sequence is obtained, you never have to go back into an animal like a poly to try and make it again. And the sequence is known, so we have the sequence in hand. So even if something happens and we lose the expression vector, we could always reconstruct it, and that's some great power to have. So ZooMabs, so let's go into the details on what ZooMabs are. So again, these are recombinant antibodies. This is a new generation of proprietary recombinant monoclonal antibodies. They're specifically engineered to combine state-of-the-art consistency and application performance. So I mentioned it's a new technology. We'll go into some of that in here in a minute. Improved consistency, very efficient design and packaging. So the marketing team spent a lot of time and effort working on those aspects, and we'll go into some of that detail. There's enhanced validation for all of these. And then there's a very robust pipeline with a lot of ZooMabs still coming out, that have come out and as well still coming. So the ZooMab technology, this kind of shows you the differences between the traditional and how the ZooMab is. So on the left is the traditional process map. So you immunize an animal, you isolate the splenocytes, and these are essentially the B cells, and you then have to do a fusion with hybridoma or cancer cells to form the hybridomas, the cancer cell lines that are used to then express the monoclonal antibody. Limited species, pretty much only rabbits and mice are used to make these. Now the difference with the ZooMab technology is we're not pulling the B cells, we're not pulling the splenocytes and having to sacrifice the animals to do this. We basically start at the B cells by purifying them from blood. So we can just take blood from the animal that's been immunized. You pull the blood that has the B cells, and through a proprietary process, those B cells are immortalized, and we can then do the screening of those B cells to identify the ones that are expressing the antibody of interest, the antibody that's specific for the target. Okay, so the screening process there is kind of similar to how the screening process with traditional hybridomas occurs. The difference there is that these are not fused with a cancer cell. So we take the B cells and we manipulate them with the proprietary process to keep them. Those cells then, once we identify which cells are actually performing and expressing the antibody of interest, we can take that cell, clone out the antibody sequence, and put it into our proprietary expression vector, and then we are producing the antibodies recombinantly. So you can kind of see on the chart on the right the advantages and disadvantages, kind of in broad terms for each of them. Polyclonals being great for species diversity, targeting difficult targets, but the specificity and reproducibility are obviously drawbacks. Mouse traditional monoclonals and even rabbit monoclonals, are not so good with the species diversity, and not as good with the difficult targets, but are obviously good with specificity and reproducibility, but the cost and the development times are higher. And then the beauty of the ZooMabs is all of them are very good. And then because we're developing these, the time and the cost to develop them is on us and not on the researchers. So, you know, you're able to just step in right there and get the antibodies, the ZooMabs from us. So you don't have to, you're not impacted by the development cost and time. So ZooMab, so the phase 1 is rabbit monoclonals. That's what we're starting with. We're starting to work on other species. We've launched a number of mouse ones. We're working on llama and goat and other species as well. And those will be coming shortly, but most of the data that I'll be presenting today is rabbit. And so some of the advantages of using rabbit as the host is it elicits a stronger immune response. You can use simpler immunogens when compared to mouse. You often get higher affinity antibodies that are produced by the rabbit. You know, there's more lymphocytes typically produced. They're easily humanized. They're all one species. They're all IgG. They produce diverse antibodies, so you do get a lot of different antibodies, and you can even target unusual structures like phosphopeptides, carbohydrates, PTM modifications. They're better suited than monoclonals for immunohistochemistry and flow cytomic applications, and produce some high-quality data that I'll show some examples of. And then finally, they recognize epitopes of human antigens that do not display immunogenicity in rodents. So the advantages of rabbits are pretty nice, and those have been well documented. So in terms of the ZooMab development process, you know, I talked about this briefly, but I'll kind of go into detail. So the immortalized B cells are screened for positive clones. Okay, so this is the testing process to find which clones, and the top clones that are tested in multiple immunoassays, the best clones are sequenced then and reverse engineered into a recombinant expression system. The ZooMab recombinant antibody is then expressed using animal and serum-free conditions. It's affinity purified. It's again tested in multiple immunoassays to ensure functionality, so to ensure that the recombinant expressed is matching the antibody that's produced from the B cell. So we do all testing at both stages to verify. The optimal concentration of the recombinant antibody is determined. And then the ZooMab recombinant antibody is actually lyophilized for long-term storage and stability. And so it's produced and shipped to customers as a lyophilized in a lyophilized format. And then, of course, there is quality control testing to ensure that the antibody is functional post-lyophilization. So we actually reconstitute and verify that every ZooMab that is produced is functional and that the lyophilization process did not hinder or impact the functionality. So here's an example of one of the ZooMabs that was produced. This is a ZooMab targeting NuN. So NuN is a marker in brain and neurons, and you can see a variety of tests here, but Western blot, NuN is known to be produced as a doublet band around 50 KD. You can very clearly see clean Western blot data. The antibody works well in IHC. So here's a mouse brain in the hippocampal region showing lovely staining. Indeed, immunofluorescence of hippocampus as well, and with a closer image of the neurons in the cortex region, as well as in immunocytochemistry using primary neuronal cultures, again showing the functionality of the antibody in multiple different immunoassays. Here's an example of a ZooMab produced for beta tubulin 3. Again, Western blot showing a nice clean band in mouth or human brain tissue. Primary cultures, or SHSY 5Y cell line, a neuronal cell culture cell line, and then as well as in IHC on mouse cerebral cortex. And, you know, again, lovely staining, lovely results in 3 different immunoassays. Each ZooMab is validated in a minimum of 3 different immunoassays. And here's a third example, a GFAP. This is an astrocytic marker, again showing Western blot, ICC and IHC and immunofluorescence. So this one even has 4 validations shown. And this is kind of typical of the data that's going to be seen for any of the Zumab targets. Here's an example of a PTM, so a post-translational modification. So this Zumab targets the antiphosphohistone H2AX, serine 139, a classic marker. And you can see in the first Western blot, this is minus and plus stimulation. So you clearly see the detection of the phosphorylation. The second image is a peptide inhibition assay. So it's a Western blot where you then do no incubation with the peptide. Incubation, the second lane is the peptide without the phosphorylation group, which doesn't inhibit or block the Western blot signal. And then the third lane is the pre-incubation with the peptide that contains the phospho at the serine 139 residue. And you can see that the antibody is completely blocked from functioning then. So this shows the specificity of the antibody for the phosphorylation site. And then it's also been tested in IHC, in testes, as well as immunocytochemistry with HeLa cells that are treated with or without staurosporin to induce the phosphorylation of histone H2AX and its nuclear signal that you clearly see in the image on the far right. So again, Zumabs can be created for PTMs. And then here's one of the mouse Zumabs that we've just launched as well, anti-Nestin. And, again, validated in Western blot on the left, in ICC and SHSY5Y cells in the center, as well as in IHC on human kidney, staining glomeruli on the left, and the negative control showing no staining as expected. So again, lots of different applications. Every antibody is tested in numerous applications. And on our website, you'll find information on every target that's produced, and it'll typically look something like this, giving background information, the specificity, details about the immunogen, the applications that it was tested on and past testing on, as well as just a general description of the target. So this is commonly found on either a C of A or on the website for every Zumab. So again, Zumabs, recombinant antibodies, the recombinant expression gives excellent reproducibility, I mentioned. It's serum-free production. There's multiple validations and characterizations, including, I didn't show it, but many of them have been tested by knockout data as well. Affinity purified for highest reproducibility and performance. Excellent affinity, avidity, and specificity. I did mention at least 3 different immunoassays. They do have high thermostability and longer shelf lives in the lyophilized form, and I'll show some of that data here in a second. And so far, we've launched over 200 Zumabs, and we have over 1,000 in development. And in fact, I can mention the COVID Zumabs actually just launched, so if anyone's doing COVID-19 studies, they would be able to use some of the Zumabs that we've produced for that. So I mentioned the lyophilization. So the Zumabs are produced and are lyophilized, and this data here shows that how the lyophilization affects functionality. And you can see lanes 1 and 2 on the left is a Western blot of one of the targets. So this was nu-N on the left. Lane 1 is the unlyophilized, and lane 2 is after lyophilization and then resuspension. And you basically see no difference in the intensity of the Western blot data. The second set of Western blot lanes there is for NMDAR2A, again showing lane 1 is not lyophilized, and lane 2 is post-lyophilization and resuspension. And again, you see almost no difference. And then on the right-hand side is some IHC and ICC staining of SOX9, another Zumab. And again, on the left are images of samples not lyophilized, and on the right is post-lyophilization and then resuspended. And you can basically see there's essentially no difference. And both of those were tested at the. So very consistent before and after lyophilization. Okay. And part of the reason we wanted to go to this lyophilization is just our commitment to green initiatives. I mean, the beauty of having antibodies, these ZooMabs lyophilized, it eliminates the use of any kind of polystyrene coolers and ice bricks and the shipping containers and the weight that is required, or that is the package weight when one of these antibodies is shipped to you, customer, is dramatically reduced. And then all the ZooMabs, I've mentioned this, but they're all are biocide, preservative, and animal free. So pure product, minimizes complications arising from potentially hazardous materials, local safety restrictions, things like sodium azide restrictions in certain locations. It's not even in the vial, so it's not a concern at all. And the recombinant platform minimizes animal involvement in production processes. So there's a lot of benefits to utilizing and shipping the products to you in this manner. In terms of the vials themselves when they show up, so the lyophilized product, will be located down obviously at the bottom of the tube, the blue little marker, but a lot of thought was put into the design and the production and the packaging of these vials by the marketing teams. Targeting and trying to generate a new generation of antibodies with superior specificity, the lot-to-lot consistency, thinking about sustainability, shipping ambiently for the environment to reduce shipping costs, obviously very stable, pure. The vials are transparent, so you're able to see that lyophilized pellet in the tube very clearly. So the vials and the labels are clear and transparent. And then a nice thing is they're actually freestanding vials, so you can actually have it standing up, or it fits in a standard rack versus most lyophilized samples do not. So this is very nice for the customer. So this is typically what the vials look like. There's an example of a label, the anti-Nestin ZooMab. I mentioned the concentrations. Most of them are at either 30 microgram per mil or 300 microgram per mil after reconstitution at 25 microliters. We in our R&D testing determine the amount that it's required, and works for the customer so that you get the proper amount, so you can just go and actually know, okay, it should work at 1 to 1,000 in Western blotting and 1 to 100 in ICC or IHC. So it really takes the onus off the customer. You just resuspend it, and then you go straight into testing. But this is typically the kind of information that you'll find on the website and or on the label. So just a couple slides on the overall antibody portfolio. We have nearly 100,000 antibodies. Obviously, they're highly cited, highly characterized. Many are award-winning. We have extensive expertise in this area with over 35 years of experience developing, manufacturing, characterizing, testing, and supporting antibody products. So, a lot of expertise in this area to help you out if you have questions or concerns. So our antibody portfolio is very diverse. We obviously have an extensive line of primary antibodies. We offer an extensive number of secondary antibodies. We offer the highly characterized prestige antibodies. Many of our antibodies are top-sided. We're now offering small pack sizes for many of the antibodies. So if you just want to test a small amount of antibody, there's that available for many of them now, and a lot of them are going to be offered in that format eventually. And obviously, enhanced validation is a key, and we're trying to note that on our website, which antibodies have enhanced validation. So in summary, our competitive edge, what we offer with ZooMabs. So, affinity purified for highest reproducibility and performance, excellent binding, affinity, avidity, and specificity, multiple application validation and characterization, analyzed in at least 3 different immunoassay applications, low bioburden development and manufacturing process. They're highly stable in the lyophilized format. They can be shipped at ambient temperature. The ZoomAbs are preservative-free, so you can use them for cell-based applications or functional assays. They're offered in multiples of 25 microliters, so you never have to buy more than what you need. They're highly thermostable with longer shelf life in the lyophilized format. And we've even got QR codes that are on these antibodies, so you can use your phone and even just click to link to the ZoomAb website to get information on any antibody without having to go to a lab, to a computer in the lab if you need information on one of the antibodies. So some of the resources that are available, so sigmaaldrich.com/ZoomAb is the site you'll want to go to. That'll get you to a landing page that's sort of the hub for all the ZoomAb information. There's brochures there. There's further articles, technical articles, and examples of numerous antibodies. Obviously, the listing of all of the ZoomAbs that we've launched thus far are located there. There's a frequently asked questions flyer that has numerous questions on it, that anything that you may have thought of. Again, if you have further questions, there's links there that you can contact tech support to get even more information or to touch base in terms of which ZoomAbs are coming out. And any of the new launches as they come out, they're coming out essentially on a monthly basis. Those would actually get posted here. And I believe there's also links to get on a mailing list there so that you could actually get that information in real time as soon as they become available. So again, ZoomAbs are recombinant antibody technology, and I thank you for your time.
Thank you, Dr. Speckmann, for that informative presentation. We will now start the Q&A portion of our webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. We already have some great questions coming in. Dr. Speckmann, what immunoassay application do you use for each of the ZoomAb antibodies?
Yeah, so for the ZoomAbs, it's target dependent. So it depends on what the actual target is that we're designing a ZoomAb for. So it's going to vary. As I mentioned, we do at least 3 for each of them, but it's going to depend on, like if it's a neuro target, we're going to do typically try and get validation for IHC because we know that that's the immunoassay that most people are going to use. Whereas if it's a cytokine, we're going to do flow. If it's, it's target dependent. If it's an antibody that we know is going to be used for epigenetics, we would almost certainly try and do chip assays on that one and validate it in IP or chip. So it's pretty much dependent. But again, we show all the data, so you'll be able to see all of that. And it is 3 applications that we target.
Thank you so much. And how do you resuspend the lyophilized pellets?
Okay, so it's pretty simple. You simply just open up the tube and you add 25 microliters of buffer, PBS buffer, or water to the tube, and you just then mix the tube gently for a few minutes, 5 or 10 minutes, and then you're ready to go, and it's ready to roll.
Thank you so much, and I want to thank our audience members for these great questions coming in. Our next audience member asks, I'm curious, how are the ZoomAb antibodies different, or why would you recommend it versus other recombinant rabbit monoclonal antibodies that might be currently being used?
Well, it's hard for me to say what the other how the other recombinant antibodies are made. I can only speak to our recombinant antibodies. I know how our antibodies are made. I know that we do extensive screening to validate them, to make sure that they're actually specific for the target, that the the non-specific binding is minimal or non-existent, that they were extensively validated in numerous applications, that there is no preservative in them, that they were made animal free. So really, it is just an antibody. Okay, so other companies make antibodies, but again, it's all of the prep work that goes into making that antibody and doing the testing on it to show that it's actually specific is the difference.
Thank you so much, Dr. Speckman. Now, how many species does the ZoomAb offer?
So currently we only offer 2 at the moment, rabbit and mouse, but we are, as I mentioned, we are working on others. We've been doing internal testing of goat, of guinea pig, of sheep, some others, llama. So, we're just not ready to roll those out yet. We want to perfect it before we launch those. But that's kind of why it's called ZoomAb, is because eventually we will have additional antibodies against different species. And in theory, it should work on most any species that we want. So that's kind of why the ZoomAb moniker is used.
Thank you so much. And we have time for a couple of more questions from our audience members. Our next one, how stable is the ZoomAb antibody after you resuspend it?
Okay, sure. So yeah, once it's resuspended, it's pretty much like a typical antibody. I believe our storage recommendations are 1 year. From that point, I would recommend that you probably handle it like you would a normal antibody. If you're going to use it within a short amount of time, 4-degree storage is fine. If it's going to be longer than that, I would probably make aliquots and freeze different aliquots just to keep longer storage time.
Thank you so much. And Dr. Speckman, we have time for one more question. Our audience member wants to know, can I use the ZoomAb antibodies with other antibodies that I have?
Yeah, absolutely. So, again, it's whether it's a rabbit or a mouse. Obviously, most of the ones that we've launched so far are rabbit. There are a number of mouse ones that have come out. It would be no different than using any other rabbit antibody that you have with another antibody and doing multiplexing experiments. Again, you would still have to test to determine whether or not there's cross-reactivity with the antibodies. But yeah, it shouldn't be any different than any of those other kind of applications. Again, the beauty of these is that they're monoclonal. There should be limited cross-reactivity. But yeah, they should work just fine. Again, you would have to test that empirically, though.
Dr. Speckman, thank you again today for your presentation and for your time and for clearly your important research. And before we go, I want to thank our audience members for joining us today and for their interesting questions. I'd also like to thank our sponsor, MilliporeSigma, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of your registration. And this webcast can be viewed on demand for 6 months until April 2021. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, stay healthy, and take care for now. Bye-bye.